Hello friends, I am Bhujbal Pratiksha. So today I am going to talking about DNA translation. So the DNA translation is the fundamental biological process that occurs within the cells to convert the genetic information stored in DNA into the functional proteins. Proteins are essential molecules in living organism to play a various roles in structure, function, and regulation of biological processes. So there are several steps in DNA translation. So for the DNA translation, first one is the transcription is occur. So before the translation can occur, a segment of DNA containing a specific gene is first transcribed into a molecule called as a messenger RNA. Then this process takes place in the cell's nucleus. During the transcription, the enzyme called RNA polymerase reads the DNA sequence of the gene and synthesize complementary RNA mRNA molecules. RNA differs from the DNA in that it contains a uracil instead of a thymine as one of its bases. Then second one is the mRNA processing. In eukaryotic cells, the newly synthesized mRNA molecules may undergo some modification before leaving the nucleus. So this modification include the addition of a 5' cap and poly A tail to protect the mRNA molecules and facilitate it to export from the nucleus. Then first step is the next one is the initiation. Once the maturation of mRNA molecules in the cytoplasm, it binds to the ribosome and cellular structure responsible for the protein synthesis. Then the ribosome starts scanning the mRNA to find out start codon. Then typically the AUG, which codes for the amino acid and methionine. This initiation complex includes mRNA, the ribosome, and initiator tRNA with a methionine. Then second one is the elongation. In this elongation phase, the ribosome moves along the mRNA in a 5' to 3' direction. Then it reads the mRNA sequence in groups of 3 nucleotides called as a codons. Then each codon is corresponds to the specific amino acid and the matching tRNA molecule carries the amino acids. Then the ribosome catalyzes the formation of bonds between the adjacent amino acid and it creating a growing polypeptide chain. Then next step is the termination. In the termination, translation continues until the stop codon, that is UAA, UAG or UGA, is encountered on the mRNA. These codon do not code for any amino acid, but it signals the ribosome to stop protein synthesis. Then release factors are the proteins that recognize the stop codons and cause the ribosome to release a newly synthesized proteins from the ribosome. The next one is the protein folding. Once the polypeptide chain is released, it often needs to fold into a specific three-dimensional shape to become a functional protein. Then this folding is driven by the interaction between the amino acids in the chain and is crucial for protein function. Then last one is the post-translation modification. In post-translation modification, some proteins undergo a additional modification after a translation, such as sugar molecules or phosphate groups, which can affect their function and localization within the cell. Thank you.